I guess first off, welcome. Glad to have everyone in the interest. And this is now, I guess, module 5 for the Snow Leopard Network's training initiative. So if you're here for something that's not genetics, you're in the wrong room. Otherwise, it's good to have you here. Just some quick Zoom etiquette. Probably most of you are familiar with at this point. Unless you're talking, to mute your mics, please. That will help make sure that the audio quality is good and we don't get any interruptions. Also, if you got the bandwidth and the connection that's supported, it's great to turn your video on so we can see who's out there and it's a bit more interactive that way. Some just formalities of how our group will be presenting to you over the next 4 weeks. It's going to mostly be a series of lectures.
You talk to Bijoy then.
We will make time for questions at the end of each lecture. You can ask your questions of the presenter, and then at the end of every day, we'll try to save a good 10-15 minutes of time for more general questions about the information that was covered. We'll endeavor to throughout some of the lectures we'll be presenting and citing some papers and all that. So every day we'll try to provide through that Google group the citations, the PDFs of the papers and any other materials that we might be presenting throughout the day, just so you'll have those for yourselves. Also to mention, Justine and Rocky reminded me of, is that if you attend all 4 of these genetics seminars over the next 4 weeks, you can attend them live in person. You will get a Snow Leopard Network certification, essentially, I guess, a nice little piece of paper to put on your wall to show that you've made it through 4 weeks of genetics lectures. And then finally, as we mentioned in the email, there's a basic, so we're presenting under the basic premise that there's an understanding of having a basic understanding of cell molecular biology. You understand heredity, DNA, and of course you have some understanding of snow leopards. So we'll be starting at a pretty low introductory level, but if you're not sure what DNA is, you may struggle to keep up. So hopefully you've had a chance to go and review some of those concepts ahead of today, but we will start fairly general. I think the basic premise of the next 4 weeks is not that you're going to be hitting the road running, doing analyses and lab work a month from now, but that you'll have an understanding of the power of these tools, how they can be used, some of the challenges, and at least be in a position to begin asking questions on how can I start doing genetic work with snow leopards. All right, so with that, I just wanted to, I guess, formally introduce myself. So I'm Dr. Byron Weckworth. I've been with Panthera as in multiple capacities for the last 8.5 years. I'm currently the director of our Snow Leopard program, director of our conservation genetics program. My background has been mostly in using molecular genetic methods in application to species and populations of concern, and working with stakeholders that have the interest in using those results from those types of studies to improve the management of the species. So, and I'll let each of our other speakers, Jan, Imogen, and Charlotte, introduce themselves quickly here. Jan, if you want to go first.
Yeah, sure. My name is Jan Janečka. I'm an associate professor at Duquesne University. And for a long time, my main interest has always been to use genetics as a tool to understand wildlife populations. And I specifically focused on wildcats. So I started out looking at ocelots and bobcats. And then, about 10 years ago, I got into trying to use these methods for snow leopards. And so it's been really fascinating, very interesting to work with everyone over in that area and just to really try to help and get information that we need on populations, connectivity, things like that. And so genetics is a very important approach in wildlife. And so it's nice to see more and more use of it on this important cat.
Imogen, if you want to go next.
Yeah. Hey, everyone. My name is Imogen Cancellari. I am a PhD candidate at the University of Delaware over here in the US, focusing on range-wide phylogeography and population genetics of snow leopards. Ironically, so I guess everyone here is connected in a myriad of ways, both the presenters as well as everyone who's participating. But from our end, so I got my start in molecular ecology via carnivore ecology. I've been a field biologist for almost 10 years, working on several different carnivore species for various projects. And as we'll be talking about this week, many of the projects that I was working on had a side project, which was a genetic focus. And that really was kind of my gateway into joining the world of conservation genetics. I've been working on wild cats for several years now. Dr. Yanechka actually sat on my thesis committee when I was focusing on bobcat landscape genetics. And I work closely with Byron, and Charlotte and I have some connections in common as well. So it's great to be here and to chat with everybody about some cool topics.
Thanks, Eugene. Charlotte?
Sure. So my name is Charlotte Hacker. I'm a PhD candidate at Duquesne University in the United States. Jan is actually my PhD advisor. And we've done a lot of collaborative work looking at population parameters for snow leopards using non-invasive genetics in Pakistan, Kyrgyzstan, and Mongolia since my time working with Jan. But a lot of my focus is in China on the Qinghai Tibetan Plateau. And I'm specifically interested in figuring out population parameters, so things like abundance and genetic diversity, but also looking at landscape genetics and in particular diet. So using molecular tools to figure out what snow leopards are eating.
Awesome, thanks, Charlotte. And next, we don't have the time for everyone to introduce themselves, as nice as that would be, but if you would humor us with a bit of information, if you go to open up the chats, the bottom of your screen. And if you wish, it would be useful for us just to know, open up, just our familiarity with everyone. If you might just take the next couple of minutes to type in a couple sentences on who you are, where you're coming from, and kind of your interest level in snow leopard genetics. Like if you want to be a practitioner, if you already are, maybe just mention briefly why you're here. Again, just a couple of sentences, nothing long, but just maybe if you want to take the next minute or two to do that, and then we can collect all of those later and just have a better idea of the audience. And then you will be able to see each other and understand who your other participant members are. So if you just want to take a minute or two to do that, it'd be great.
Hey, Byron, can I ask another silly question? Kostov here.
Yes.
Are you by any chance recording it, this session?
Am I recording? I think it is. It looks like I have the option to stop video, which would suggest that there is video being taken.
We're recording for the cloud in SLN, Kostov.
Oh, perfect. That's
yeah.
great. Thanks, Justine. Thank you.
No worries.
Yeah, I'll have to kind of once in a while rush to manage a storm around me, but yeah, that's why I just wanted to not miss out anything here.
That's a good point, Byron. If I can just mention, Reki put in the chat that we are recording, but it's probably already lost. So if anyone has any objections to the recording or have anything concerns, please write to Reki. And then she'll take note. Thanks, Kostov. It's good to highlight that.
Thank you.
Yeah, I forget. Sorry, Justine. Maybe I was supposed to mention that at the outset. I'm doing very good in my hosting duties.
You're doing fabulous.
Okay, I don't have the chat in front of me, but I assume that people are hopefully providing a bit of info. Definitely throughout, if you haven't had a chance to do it yet, there's the chat. You can add it at any time throughout today's lectures, and we'll pull those later. Just so we have a basic idea of who the audience is, and we'll have some more interactive polls and things later, just to learn more about who is listening and what your interests are. That will help guide some of the some of the discussions, particularly on the last day. But getting to a bit of an overview of the next 4 weeks. So today we're going to have 3 main parts. I'm going to start off with providing some just basic overview of conservation genetics and the opportunities and needs for snow leopards to kind of set the stage of why hopefully a lot of you are here and what your interests are. And then we're going to start at the various sort of beginning levels of things to understand what are we talking about with regards to genetics and the markers that we're using for the purposes of these various conservation needs. And then that will be presented by Imogene, and then Charlotte's going to talk about some of the difficulties and challenges involved in non-invasive genetics. Like you're starting off with a realistic set of assumptions on how this can work and how you need to set yourself up for success. In the subsequent weeks, we'll get into, if you could only attend one of these 4 days, I would say next week, when we get into sampling design, sampling and lab etiquette. Those are absolutely critical to having a successful project. If you will have good quality data and have the type of data to answer the questions that you're interested in, you need to have good sampling design and really good, elegant sampling and lab etiquette. That will be followed up in the 3rd day by species individual ID, to understand how we do that. Getting in a bit to not really describing how to do the analyses, but describing what analyses bring you to analyzing the raw data and putting it into context for some of the questions you might have, with a focus on individual species ID, getting at counting cats, how we can use these methods for getting at diet, as well as some ecological landscape modeling concepts. And then the last day, we'll have a lecture on next generation sequencing, which is essentially the cutting edge of molecular methods that we might apply and want to apply to snow leopard work. And then we're going to open that up to more or less a panel discussion on conservation genetics and practice to involve all of you a bit more and better understand how we can set up all folks throughout the range countries to have success in applying and using these methods. Some introductory terms, just so we can all kind of maybe start on the same premise. If you were to do a search for define conservation genetics, you're going to find vastly different, slightly not completely different, but much a large variety in wording for what are conservation genetics. So in the context, for me, and if you were to ask Imogene or Charlotte or Jan, they're going to probably state this slightly differently. But from my standpoint, what we're talking about when we're considering conservation genetics, it's an interdisciplinary field that aims to describe and understand the genetic dynamics of populations, populations being a bit of a loaded term, depending how you're measuring what a population is. Anyways, of these units of conservation concern in order to inform management and policy action to prevent extinction. So that's what we're doing when we're doing conservation genetics. And then the idea, we're going to get a lot more details as the day goes on, but non-invasive genetics versus conventional. So probably, if you've been paying attention to articles or the science to date on snow leopard genetics, it's mostly non-invasive. But what we usually mean by conventional, what that is an alternative to, is conventional would be having muscle, hair, or muscle or skin or some other tissue sample that you actually have to have an animal in hand to collect blood, of course, like blood from captures, something like that. Non-invasive genetics, on the other hand, is something that we get in a sort of a forensic style sample, hair, scat that we collect without ever actually having to see the animal. And that works really well, obviously, for snow leopards. It works well across the whole host of other organisms. But the nice thing about it, there's a lot of negatives to that we'll get into. But the positives of that are you can get a lot of data, a lot of samples that actually happen to see the individual, which for snow leopards, saying is pretty useful. You don't see those a lot. And also you're minimizing the effects of capture or tagging animals. You know, it's passive, and for the most part, you're not having an impact on an individual or a population of individuals. So with that in mind, just here's a couple slides to get our minds thinking about what's the use for these and why genetics is important for our work on snow leopards. As I think we all can appreciate that of all the threats and risks facing snow leopards, that all those challenges are exacerbated by a lack of knowledge. And that lack of knowledge extends from everything from understanding just simply where are they, their range, certainly the numbers. If you were attended any of the pause sessions earlier this month or last month, late last month, getting at connectivity, it's animal that's widely distributed across these naturally fragmented mountain ranges. Connectivity is important. But even basic tenets of ecology and evolution, we're still left wondering and is left unknown for snow leopards as compared to other large charismatic species. And as it turns out, that there are genetic research methods and tools that can address all of these knowledge gaps, and that can be used in ways that are, as basic as survey and monitoring efforts, but also to apply to hypothesis-driven and application-driven research projects. So this is where I like to sort of demonstrate that across all the big cats, proportionally, if we consider where snow leopards are at, given that, I've just demonstrated or suggested to you that genetic methods are, what we need to address all these knowledge gaps to the snow leopards. Yet for snow leopards, as compared to other big cats, proportionally, much fewer studies have been done. So that's just contributing to this massive knowledge gap, as we're not even haven't yet sort of harnessed the power of the best tools that are out there to begin addressing some of these questions. So there's huge, what this means is there's huge opportunity that anything you do with snow leopards using genetic methods is going to be important, is going to add significantly to our knowledge base, because our knowledge base is quite small as compared to, in this case, all the other big cats. So just getting into some examples of these, of how non-invasive genetics can help, just in some of the basic survey and monitoring efforts. Definitely, you can go out there and picking up scat, hair samples. It's mostly scat with snow leopards that we're using. If you combine this with your camera trap detection and your camera trap grids, really increasing your opportunity to detect the species. Also, you can do this a lot more quickly than, for example, camera traps, where you can go and do a one-off survey. If you're out in a new landscape, it's never been sampled before, no one knows are snow leopards or not, you can simply do some transects, get some samples, bring those to a lab, and you'll know without ever having to return were there snow leopards there. Versus if you were to set camera traps, you have to set those up, you have to monitor those, maybe replace batteries, change memory cards, go back and forth at least once or twice to collect the cameras and get the data. So that you can mobilize these methods pretty quickly to do quick surveys just to get at presence absence, getting at numbers. In addition, so this is largely in comparison to maybe traditional camera trapping methods. So in addition to getting, all right, there's a snow leopard here, yes or no, presence, you can also get at sex. We can use molecular methods to sex where the sample came from. It's pretty difficult in most cases with cameras. We can do, spatial recapture, estimates of abundance with your detections, individual detections from the genetic methods. We can get an effective population size, which in many cases, particularly in species that have small population sizes, low density, and are fragmented, I would say is an even more important metric than necessarily census size. That's something much easier to do with genetics. And then individual ID is far superior to cameras. When you look at probability of identity, so probability of identity is the probability that any two randomly sampled individuals in your sampling unit are identified as the same. That probability is really low. So essentially, this is think of this your statistical power of counting individuals. So the probability with a set of like 12 microsatellite loci is 2.35 to -7. If you have a general a moderate number of SNPs, that probability is even lower. So the statistical power of these methods is pretty incredible. And when done right, understanding the assumptions and biases is going to be pretty reliable. The non-invasive genetics as well provides some nice uses with regards to combating other threats. So illegal wildlife trade is huge and probably an increasing threat for snow leopards as the demand for tiger bones and other big cat bones as replacements continues on. And so with these methods, we don't necessarily, you can have a product that maybe you can't identify what that species that came from, but we can use genetic methods to figure out what that is. And another sort of cutting edge method that's developing right now is eDNA. That's where we can detect the species without even a scat or a hair sample, where we can sample, you just think about sampling along ridgelines where you think snow leopards are maybe traveling and picking up DNA that's just in the environment and not necessarily attached to a traditional sample. This is a technology that's been used, it was developed in aquatic systems, but it's rapidly being transferred in terrestrial systems with really amazing impact. And I meant to provide a citation here, but I'll send it along in the Google chat of a great study that came out earlier this year comparing species detections with camera traps versus eDNA methods. So where they had camera traps set up at a regular grid and they're sampling soil along the game trails and getting DNA from the soil of all the animals that are walking on that trail. The results are pretty amazing. So eDNA is another sort of rapidly developing technology that's going to help us with our survey work. Getting into some of the research needs. Right here. So I think we could all, if we had a discussion, maybe reorder or add or change some of these priorities. I think that with snow leopards specifically, given the knowledge gaps that we have and the importance of filling those gaps to better inform conservation, that starts with just understanding patterns of the range-wide scale of diversity and divergence. So getting at subspecies or ecotypes or these large core scale definitions of evolutionary history. And then also using that to inform how climate and predict how climate change might impact some of those things. And I'll get into some examples in a minute. Getting into understanding, of course, the importance of corridors, impacts of landscape on the species, and especially with regards to population and population dynamics, describing and allocating, drawing your lines on the map that define conservation units at which management and policy strategies will be implemented. And also getting into how we can use genetic methods to better understand disease ecology and epidemiology. I know across much of the range, domestic dogs are an increasing issue for lots of reasons, both ecologically, but also as vectors for disease. So I think it's important that we keep thinking about disease a bit more as well. But so some examples here, just how you might apply some of these. So this is tigers across Asia, where they're left. And you can see, and again, I'll provide the paper, the citation here. The distribution of genetic diversity at a core scale, sort of a subspecies scale for tigers, shows that there's these distinctive subspecies, Siberian, Chinese, Malayan, Sumatran, and Bengal subspecies of tiger, but that a considerable amount of genetic diversity. Each of these circles is a unique haplotype, unique sort of genetic signature of the species, that a large proportion of that diversity is found within India. So that as you're designing your sort of range-wide species conservation goals, the emphasis on the importance of India as a sort of as a source of tiger genetic diversity, can't be lost in those management conservation strategies. So getting into a paper that came out a couple of years ago on the impact of climate change on snow leopards. So this is snow leopard range and the predicted changes in their range with the various climate scenarios going back to the last ice age and then projecting forward into 2070, and better understanding what has been the impacts of previous large climate change events on snow leopard distribution and the related shifts in genetic diversity, like identifying in these hashed lines, these large refugia. So we can go and genetically test this model to say, are these areas that have served as refugia in the past, and then consider those in the context of what we're predicting will be refugia into the future, and then defining that through genetic means, and then developing large-scale strategies to make sure we're preserving those areas of important long-term range genetic diversity. Again, considering at, again, this coarse scale, gene flow and connectivity across the range. This is from a paper earlier this year defining priority regions for snow leopards. So the areas in red are those that are deemed to be really high quality, really good core snow leopard habitat. And we can see how those are distributed across the range. And this, again, is a model of habitat. We would like to go in and test this model and say, do we see among populations distributed across snow leopard range, are they connected in this way? Are these areas, these pinch points of particular relevance in keeping large-scale connectivity in play, and then designing, again, the way that we maybe implement new protected areas around emphasizing these important pinch points and areas of connection across the range. We can take this to a finer scale. So this is bringing it back to tigers and looking in central India among protected areas in tiger. What this paper have done is they've used least cost paths and current flow methods to look at corridors among protected areas. And again, think of this graph or this figure as a hypothesis. This is based upon habitat needs of tigers, how they predict the connections among the protected areas. Where genetics comes is it comes in and tests us and say, are we predicting areas of connectivity among those protected areas for tigers correctly, or some of these areas we think are connected are not, or maybe some that we don't think are. So genetics provides a great tool to then test a lot of our habitat modeling and a lot of these connectivity analyses that, it comes down to it. You can predict it all you want based on habitat, but what really is important for connectivity is the exchange of gametes, gene flow. If individuals aren't moving between habitat patches and successfully reproducing, then your connection is not good enough. Just pure use of it by itself is inadequate. So we really need to make sure that as we're designing these linkages among our protected areas and habitat patches, that they're facilitating gene flow. That's ultimately the most important reason for developing connectivity. And then finally bringing it to a different part of the world. This is some work done on caribou that I had done years ago, but defining conservation units. So this again can happen at various different scales. So this is a very fine scale. In the earlier slide, I mentioned defining units based maybe upon subspecies, which would be important for IUCN designations. But on the ground management wise, it's often a much finer scale. So just pull you into this real quickly, that these are caribou populations across an area of Western Canada. And they're currently managed as 18 different units. So each of these polygons has a different management strategy from the other. Yet, if you were to look at what are the actual genetic relationships, what are the meta-population dynamics among these different populations, you see that they probably, versus 18 different units, should be managed as 6 different units. And that would be much more effective and efficient management, both we think about the application of resources, monetary resources, but also time and effort on the ground. So lacking the genetic information to describe the gene flow and relationships among the populations, you're stuck managing this way. If you have that data available and you analyze it correctly, you can really streamline your management strategy and ensure hopefully greater success in meeting conservation outcomes. And then finally, again, I think there's a lot of application of disease to using genetic methods to get at answering all sorts of questions on disease. And again, this is something we could spend an entire module just talking about this, but really getting at rates of infection, susceptibility to infection, how infections are spreading across landscapes. We can do a lot of that, predict that, model that, and then test those things using genetic methods. So, and just a quick conclusion before I hand it off to Imogene's next. is first, beautiful snow leopard pictures. Always have to credit those where they come from, but also just point out that from scat, from all you've been at who've done snow leopard work, and you see the piles of scat everywhere. It's an incredibly valuable resource, and all these things I've mentioned, we'll get to some greater detail on some of them. Others, I can pass along papers, we can have discussions outside. But there's an incredible wealth of knowledge wrapped up in these scats, and that I look forward to talking more with all of you as we move forward in the coming days. But for now, I will hand off to Imogene.